Assalamualaikum, this is PharmapHila with a new video of anti-diabetic pharmacology series. This is second video of this series. So here I am discussing with you the insulin. Insulin is a small protein, innocent protein, which have a major role in our body with a molecular weight in human 5808. It contains 51 amino acids arranged in the two chains A and B, linked by disulfide bridges. Proinsulin, a long single chain protein molecule, is processed within the Golgi apparatus. As you can see in the childhood, in the animal cell, there are the Golgi apparatus, amoeba-like structure inside the cell. These are the Golgi apparatus. So within the Golgi apparatus of the beta cells, the proinsulin process and packed into the granules, where it is hydrolyzed into the insulin and the other component called C-peptide, or it can also give as a waste product, like a waste product of the 4 amino acid. By the removal of the 4 amino acid, we get what? Insulin. And from which product or from which reactant? That is a proinsulin. Okay, this is a innocent structure of insulin. These are the two chains A and B and insulin. The orange shaded structure is insulin. It have two chains A and B. As you can also see, there is a disulfide bond. In the Golgi apparatus, they break into the insulin and the C-peptide by the removal of 4 amino acids. So what you have to learn in this picture or in this story, that insulin is 51 amino acid arranged in the two chains, that is A and B. That's it. Now what next? Insulin and C-peptide are secreted in the equimolar amount. That means that their molar concentration and their molar are same. In response to all insulin secretagogue, granules within the beta cells store the insulin in the form of the crystal consisting of 2 amino of zinc and 6 molecule of insulin. The entire human pancreas contains up to 8 mg of insulin. 8 mg is the huge amount, representing approximately 200 biological units. So we are thankful to Allah that we already have endocrine system that give us 8 mg of insulin. Alhamdulillah. Insulin secretion. Insulin is released at the low basal rate and at much higher stimulated rate in response to the variety of the stimulus, especially the glucose. When we eat the food, it is a stimulus and our body releases insulin. Other stimulants such as other sugars like the mannose, amino acids, hormones such as the glucagon like peptide, it has a particular role in the diabetic mellitus, as you can learn in the next topics. Glucagon, higher concentration of the fatty acid, and the beta adrenergic sympathetic activity are recognized. Stimulatory drugs are sulfonylurea, meglitidenide, natiglinide, isoprotenolol, and acetylcholine, and these are some inhibitory signals or hormones including insulin itself, leptin, alpha-adrenergic sympathetic activity, low concentration of fatty acid. Inhibitory drugs include diazoxide, phenytoin, vinblastine, and colchicine. Okay, this is an structure. What does this structure want to tell? The secretion of insulin. In the above side of the picture, you can see the potassium channel. Potassium is the inhibitory. Sulfonyl drugs block the potassium channel. What happened? When the potassium channel block, these are the inhibitory, depolarization occur and ultimately stimulatory signal enhance. Then when the stimulus get our body, it will release the insulin. At the right side of the picture, there is a calcium. What is a calcium? Calcium is the inhibitory hormone or the inhibitory signal. Calcium channel help in depolarization, open the channel, calcium goes inside, it will release the insulin from the beta cell through the process of the exocytosis, and insulin come and attack to the targeted organ, decrease the blood glucose. Then what you can see at the left side of the picture, it is a glucose transport. Glucose goes inside the cell by the glucose 2 or the glucose transporter. So the metabolism, ATP produce. ATP, what does this ATP cause? It depolarizes by the release of potassium, closes the channel. When the potassium left the cell, it will depolarize. Potassium is basically inhibitory and calcium is potassium is inhibitory and the calcium is excitatory. We do not need potassium in the cell for the release of the insulin. We need calcium in the cell to the release of the insulin. So this is the basic secretion of the insulin. So for today, this is enough. For the further next lecture, I will define you more basic concepts about the anti-diabetic drugs, insulin and its organ effects. Thank you so much. Till that, Allah Hafiz.